Thank you, Petra, and thank you, Sarah and Andrea, for stimulating and thought-provoking talks. Together with one or two other members of the audience, I'm in the category she's trying to sort of slow down the aging process in it. So the sooner she comes up with some successful answers, the better. So we're now moving on to this final session, addressing some of the challenges of the food and health landscape. Ramnik, Xavier is the first speaker. He already got a big introduction from Petra to start with, so I'm not going to repeat it, Ramnik. He also has a little bit of a challenge for travel, so we're going to change the format. And when he's finished his talk, we will then have the question and answer on his talk before we go on to the final talk from Alex. Okay, so over to you, Ramnik.
Okay. Want to thank Petra and the organizers for putting this meeting together. I always learn during these NINS meetings because the topics are very broad and often they go outside my area of comfort. So what I thought I'll try to do is give you sort of a perspective on sort of various aspects of how, so I was asked to connect the diet, the gut microbiome, and health. So the topic was given to me. And so what I'm going to try to do is try to give you a couple of examples, but then end with a vignette. So broadly, as we've heard over the last 2 days, we are what we eat. The nutrition we take makes us, builds our protein, our fat, our carbohydrates, our muscles, and so forth. But when we think about the landscape of food, we think that that's the largest component, but then you start looking with the microscope and you realize that the microbiome is equally large. So there's this competition between, we are what we eat and we are also what we contain, and that I'm going to refer as the microbiome. So a few facts. So the human genome consists of about 20,000 protein encoding genes. These genes encode for proteins that are units of function within an organism. And the genome is the blueprint of our metabolism, our immunome, or whatever you want to define it as. Whereas the microbiome is a lot more complicated. So there are about 4,500. So it becomes a counting game of how you count bugs. How many bacterial species are in the gut? They contain about 100 to 1,000x genomes. So it significantly multiplies the human genome, and then the microbial genes extend host genomes and provide unique functions. So I'm going to suggest that there are probably this sort of explosion that's going to happen. Nestlé has been sort of dabbling in it with a little bit, where there is likely to be about 200,000 new chemical entities that the microbiome is going to add to our circulation on a regular basis. So what does the microbiome do? It modifies our diet. We've heard a couple of different examples. It modifies the drugs we take. It increases or decreases the bioavailability of these drugs. And then more importantly, it plays an important role in host-derived metabolites. So when I was in medical school, the textbooks of medicine said there were 2 primary bile acids and 2 secondary bile acids. Now there are 100 different bile acids, so suggesting that the microbiome can modify the primary and secondary bile acids to be many other components. But the way I want to try to frame my talk is to basically define how does human physiology get altered. So we've heard a lot about how the diet impacts it. We've heard about exercise that impacts it. I'm going to tell you a little bit about how the microbiome impacts it. But then there's obviously circadian rhythm and other factors that play an important role. As Andrew alluded to, the first few time span of life within the utero and so forth. So the body needs to coordinate these activities to maintain a healthy physiological state, and hopefully I'm going to convince you that there are places to start. So this was a review that appeared in the New England Journal of Medicine earlier this year, and I said to myself, what's the evidence for these various dietary regimens that have been proposed? And how are these dietary regimens built? So what are the building blocks that add to it? Obviously, this is a biased view of the 2 authors who wrote this paper, but I think it summarized well how these various dietary factors are captured. So what I'm going to try to pick on one or two of these diets rather than go through them individually. So what about ketogenic diets? So they basically produce ketone bodies. They are composed of low to very low carbohydrate content, but no restrictions in lipids, proteins, and energy intake. So some of the points that Sarah alluded to. The initial therapeutic focus when ketogenic diets were introduced was to basically control weight loss and manage drug-resistant epilepsy. It's been shown to reduce cardiovascular risk factors, effects on overweight, obesity, and type 2 diabetes. And more recent studies have suggested that it could also play an important role in cancer. But I think the real evidence-based finding of ketogenic diets is its impact on resistance to seizures. That's where the ketone bodies have been connected to the neuronal synapses and shown that you can control it. So how do we extend this? So Xian was a recent postdoctoral fellow in the lab, and when she was a graduate student, she basically asked, how does ketogenic diets influence the immunology of the gut? And what she was able to show elegantly was that low carbohydrate, high fat ketogenic diets. So if you looked at that New England Journal review, there were many flavors of ketogenic diets. They affected the gut microbiome. Ketogenic diets that made ketone bodies shifted this molecule called beta-hydroxybutyrate. And beta-hydroxybutyrate affects how T cells evolve or respond to health and disease in the gut. So if you take beta-hydroxybutyrate and then ask, what does it do to other components? So there was a recent paper published in Nature where they were able to show that ketogenic diets, particularly beta-hydroxybutyrate, so you're measuring diet to a molecule, it affects tumors in the gut. And the data was quite striking in mice. And then they came to Switzerland and looked at a very large clinical cohort and showed that the presence of beta-hydroxybutyrate in humans also had an impact on colorectal cancer. Now, there are mechanisms behind it. The authors went on to show in organoids how beta-hydroxybutyrate affects the potential for an epithelial stem cell to become a cancer cell. And so there was what I call diet to molecule to mechanistic evidence. Now, what about a second example? So we've also heard that fiber generally does lots of good things, but if you go back, and I was part of a study done at University of Pennsylvania in 2021, where there's a physiology unit where you can lock in 24 individuals and measure the food they take. They're fed from the same kitchen. They cannot leave the premises. And so you can then ask, how does diet influence various measurements? And what this study showed was clearly the fiber diet alters the carbohydrate, the firmicutes especially alters carbohydrates and amino acid gut metabolites, and diet-based microbiota metabolome, so the stool metabolome has an impact on circulating metabolome. So you can now extend what's measured in stool all the way to circulation. Now, having said that, fast forward to 2024, they basically asked a very simple question. When you put patients on these healthier diets, there is a condition called irritable bowel syndrome. So this is a broad condition that affects many individuals. And if you do a poll in this room, at least 20% of those in this room have had symptoms of irritable bowel syndrome. So this is very different from inflammatory bowel disease. The finding of diet and its effect on this healthy condition depended on the presence of blastocystis. So there was now a microbial connection to how diet impacted health. And what I liked about this study was that they could then take these observations and go back to reductionist model systems and show that this microorganism had an effect on how diet plus a microbial intervention altered a clinical outcome. And it also had effects on cardiometabolic outcomes and so forth. A second question. So for the last 100 years, there's been this ongoing debate in the literature in physiology that exploiting diet and nutrition plays an important role in tissue regeneration. So we heard a brief introduction from Andrea. And so what do we know? So 400 years ago, Luigi Cornaro said, how can you live to 100 years? By dietary restriction. And for the past century, Number of investigators have gone on by working with model systems and human systems to try to extend this life. It's worked very well in yeast and small animals. But what we've begun to learn is that there is a cap. You basically cannot increase your life beyond 125 years. And so where does diet impact in a life extension study? And so a few years ago, we asked a couple of simple questions. So when we talk about diet, very few physiological measurements are done. So in this particular experiment, we asked the question, when you expose individuals, healthy individuals as well as mice, to a mixed diet that contained equal amounts of protein, carbohydrates, and fat, what happens to digestive enzymes? And in this particular example, we measured the production of trypsin. So if you look at the textbooks of physiology, trypsin is made in the proximal gut through the pancreas. As a chymotrypsin, it gets activated. But trypsin needs to be inactivated before the next meal for the next spurt of trypsin. And so very little time was spent on how to inactivate trypsin. And what Kenya Honda we were able to show was that there is a bacterial species in the terminal ileum of healthy individuals that has an enzyme that can convert trypsin to an inactive trypsin, thereby now preparing the pancreas and the proximal gut for the next burst of digestive enzyme production when you see the next meal. So adding some complexity on how physiology regulates human nutrition. What about the stem cell? I told you about this observation 400 years ago and how for the last 100 years everybody's been trying to extend our lifespan. And this stem cell concept is true whether you look at a muscle stem cell, a gut stem cell, a hematopoietic stem cell. And what's emerged is that various dietary interventions alter stem cell activity and biology. So I'm summarizing work. So this was based on some early work that we had done, where we had shown that the stem cell is a niche environment that is protected. Based on our observations, various labs have gone on to show that ketone bodies, alter stem cell function. They maintain homeostasis. A high-fat activated fatty acid oxidation also alters stemness. And then more recently, the expression of certain genes on the stem cells is also altered, and thereby, when you don't have these protective functions, you basically increase the risk of cancer. Now, if you look at cohorts in Europe, in the US, there's this marked uptick in early onset colon cancer. I've seen a patient at 29 years old who presented with colon cancer with no family history. And based on the findings, the recommendation for diagnostic or screening colonoscopy ages are moving up. So right now it's at 45 in the US, it's expected to go down to 40. So could nutrition play an important role? And a recent elegant study that was published in Nature last week basically did a clever experiment. They ended up fasting these mice and then refeeding them and asked what happens to stem cells. And what was pretty amazing was that they went on to identify the signaling pathway. So it activated mTOR activation, it increased polyamine synthesis, and polyamine was the metabolite that was involved in EIF5 alpha phosphorylation. But what was striking is that fasting and refeeding signal was critical. So if you had a healthy stem cell, fast refeed cycle maintained a healthy stem cell. But if you were unlucky and you carried a mutation in the APC gene, so this is the APC adenomatous polyposis coli gene, that is a precursor mutation that increases the risk of colon cancer, the fasting refeeding cycle activated that APC mutation, and these mice ended up getting colon cancer a lot earlier. So you need to have a healthy genome to really benefit from the dietary interventions and the signaling paradigms that might emerge. So in the second vignette, I've tried to tell you how we can use diet, the microbiome, and then drill down to a specific cell, a stem cell, and tell you what might be the mechanistic pathways that might be operational. And as we go on to advise the consumer, we can start to think about what's the evidence that exists. So let's move to the extremes of age. So we learned from Andrew the effect of the methylome. We, over the last few years, have basically asked questions about early life events that shape the microbiome. So we were able to show with others that delivery mode has an impact on what the microbiome is, vaginal delivery versus a C-section. The microbiome becomes adult-like in approximately 3 years of age. Generally, human genes go vertically, so they go from mother to child or father to child. But bacteria are unique that they can also go horizontally, so they can go from the bacteria to the child's bacteria and then introduce gene changes. And a lot of these, so these are these mobile genetic elements, and I'll talk to you a little bit about it, but a lot of the microbiome studies are done in the West in early development. And with Nestlé, we were able to look at a cohort of individuals in Bangladesh doing the same trajectory, and the learnings from that were few. So getting back to the mother-child connection with the microbiome. So in this particular experiment, we enroll mothers in Helsinki in the second trimester of pregnancy and asked what happens to the microbiome and the metabolome. So I told you the microbial metabolome is reflected in the plasma metabolome, and asked what happens from the mother to delivery and first 3 months after delivery, and what happens in the child. And you can begin to see that there's this huge explosion of metabolites that appear in the child that were never made by the mother. And following studies have shown that these metabolites are extremely important in cognitive development and immune development. So suggesting that understanding these trajectories are important. But one of the key lessons that we learned from this is we were able to identify microbial genes that jumped from the mother to the child 3, 6, 9 months after delivery, probably making the case for maternal bonding. And the genes that jumped from mother to child were genes that led to the production of metabolites that controlled immunity and cognitive development. So suggesting that there's a lot to learn from these types of observations. The study with Nestlé was to look at children in Bangladesh, and one of the striking observations was this appearance of this transitional clade that's shown here. Of B. longum that appeared only in the children in Bangladesh. You look elsewhere, in children born in the US or Europe, this clade is not seen. You could find this clade in children born in Peru, suggesting that there's an importance of studying microbiomes geographically. And if you can look at the literature of microbiome that contributes to the global literature, you can look at what happens, the contribution from the developing world. It's a small slither of data. In collaboration, what we've been able to show is that this transitional clade is the best utilizer of human milk oligosaccharides. Furthermore, this transitional clade also makes a molecule called pipercholic acid, and pipercholic acid is a fetus lengthening metabolite. So it basically increases the linear length of the child, suggesting that metabolizing human milk oligosaccharides at the right time results in a molecule that makes birth weight more or less stable, but the length of the child being different. So in the second example, I've told you about the childhood. What about the extremes of age? So I had this opportunity with collaborator Kenya Honda in Japan, where we were able to visit Okinawa. So Okinawa has the highest number of centenarians. So we were able to study over 100 individuals who were over 100 years old and asked in the household, so these were individuals who were over 100, their children, their grandchildren, and the great-grandchildren were in the same house, and we studied the microbiome in these children and asked what might be different. And since then, we've also been able to confirm these findings in Sardinia. You can see the appearance of these yellow and red signal that appears in the centenarians. And what we identified was that in the centenarians, there were 2 bile acid modifications, the iso-LLCA and the oxo-LCA, modifications of these secondary bile acids by bacterial enzymes that in fact played an important role in protecting you from infection, but more importantly expand the number of these regulatory T cells. So this is an example of what human physiology is teaching us by looking at the extremes of age. Told you about early childhood. Now I'm telling you about centenarians, and these are what I call field experiments that have told us how we should start rethinking about nutrition, diet, and the microbiome. I'm going to end by a vignette. And so in 2019, I had a graduate student at Harvard who came to my lab and basically showed me this picture. So there was this observation in the 1930s that there was something in stool that converted cholesterol to coprestanol. And he basically told me, I want to work with you, but I have my project. You cannot give me a project. And I said, I have no clue about cardiovascular biology. If you can convince me in the next 2 weeks, I'll take you on as a student. What he was able to do was basically identify the enzyme that's involved in bacteria that can convert cholesterol to coprestanol. And we've since then gone to the Framingham Heart Study and looked at individuals who have high cholesterols and low cholesterols independent of statin use, and showed that the presence of this enzyme is predictive of cardiovascular health when it comes to lipidomics. So the way Doug Kenny went about doing it, so obviously as a quick summary, there are lots of interventions that decrease total cholesterol. Most of them are medications that you have to take on a daily basis once you have a high blood cholesterol. And maybe there might be this opportunity of finding a microbial intervention that might reduce the need to take increasing doses of cholesterol. So Sarah showed us this curve about how cholesterol changes with age, and maybe that peak or that uptick could be reduced. So the way Doug Kenny went about doing this was he wanted to find out what were the genes that were responsible for this conversion. Could you identify the bacteria that encode this pathway? Could you then understand the consequences of this bacteria in cholesterol metabolism, and what are the effects on serum cholesterol? Because at the end of the day, what you want to know is what happens in circulating cholesterol, not stool cholesterol. To do this, he built an assay that allowed us to measure quantitatively, both in stool and blood, the levels of cholesterol and coprestanol. And so that you could take any biospecimen and say, how much cholesterol do you have, how much coprestanol you have. Using that, we then asked the question, could you identify individuals who encode for this enzyme, or those individuals who lack this activity and altered measurements of coprestanol and cholesterol. To do this, he went and built a cohort that extended all the way from the United States to Netherlands to China. So he got a very broad spectrum of individuals and asked the question, is there activity in the bacteria that could convert cholesterol to coprestanol? I'm going to summarize. In the 1990s, there was an observation in a journal that's not read very often, that there was some activity in hog sewage that could convert cholesterol to coprestanol. So what he did was to go and identify the bacteria that's dominant in hog sewage, sequence the entire genome of that bacteria, identify the potential enzyme, and then asked, does that enzyme in fact convert cholesterol to coprestanol? And the answer turned out to be true. We were still very far from identifying a human bacteria that can do this conversion. And then he used computational biology to try to identify these cholesterol dehydrogenases and was able to identify 25 different bacteria that had these enzymes. But we could not pinpoint to the exact taxa because a lot of the microbiome is dark matter and you can only show disease activity. What he then did was to show that this was clearly enzymes. He basically took the ECOP 170 that came from the Eubacterium coprestanoides and the 25 associated human bacteria and made mutations within the binding pocket and was able to show as you make mutations, you lost that conversion activity. So in this example, what we've been able to identify is a series of bacterial enzymes that are as good as taking lovastatin, a statin, or PCSK9 to convert cholesterol to coprestanol. So the market share here is thought to be something like $2.58 billion, and this is something like $13 billion. So now we have a bacterial enzyme that can convert cholesterol to coprestanol as good as the currently prescribed drugs. So what I'm getting to is more sort of to highlight how we get to mechanism and moving from association studies to figuring out how does something affect the diet. So summarizing what happens is that cholesterol is made in the gut, comes from your diet, comes from various factors. Most of it gets reabsorbed. If you now can convert cholesterol to coprestanol, coprestanol cannot be reabsorbed. It's then filtered in your stool. So you basically end up creating a sink that dumps the cholesterol without getting reabsorbed. And this is what I showed you based on data. So we were lucky where we were able to gain access to the Framingham Heart Study. This is 75 years of people followed over time where their cardiovascular status has been measured. Now they're in their 4th generation. And we were able to collect data from something like 1,500 individuals. We got 250 measurements of their clinical data. We got measurements of their lipids over time. We've got measurements of all the medications that they were on. And asked the question, could you then identify bacteria in these individuals that were associated with this low cholesterol reaction? The answer is yes. So we were able to identify this bacteria called Oscillobacter that was highly prevalent in this Framingham Heart Study. We've since confirmed this in other communities with heart disease and showed the presence of Oscillobacter is associated with decreased cholesterol levels. These individuals are not on statin drugs, and they have a baseline cholesterol. So in addition to cholesterol, it also affected triglycerides, because some might say that cholesterol is a marker and you need to have a much larger profile. And then basically, we then took this bacteria and did metabolomics, and we could see that if you had the bacteria in the presence of cholesterol, it could convert cholesterol to these products. Now, the real challenge was when we went and inspected the genome of the bacteria, we couldn't find this ISMA enzyme. So by sequence identity, we couldn't connect the genome of Oscillobacter to the bacteria that I showed you. And this is where AI and machine learning came into play. So one of the ways of doing this is to use protein language models to detect homology. And so briefly, sequence identity is correlated with structure. And so just like doing sequence similarity, one can do structure similarity. The attraction of structure similarity, it's much more conserved than sequence similarity. Then Chen Hao in the lab built language models to recover this structural similarity. And guess what? He was able to show that this enzyme has perfectly folded, just like the other enzyme that we identified in hog sewage that was within Oscillobacter. So if you stretch it out, you can't see it. You allow it to fold, the fold then shows you that there is an active pocket. And so using this approach, now we could basically show that in the 3 different types of Oscillobacter we found, we could find this enzyme activity by structural homology. And since then, we've done various mutations to show that structure predicts form and function, and this enzyme, in fact, can convert cholesterol to coprestanol. So I think there's going to be a huge impact on AI, machine learning, language models that's going to impact the food science, nutritional science industry, but also how various components are affected. So the final experiment we wanted to know was can you then show that this is true? So we took labeled cholesterol and fed them to these Oscillobacter bacteria. We were able to identify bacteria that had the enzyme and bacteria that didn't have the enzyme, and we can now show that, I think something happened to this slide, but basically you could show that you had the ability to convert. Now the benefit of this was we also now found an enzyme within this same bacteria that can make active vitamin D. So there is an enzyme that can basically take and convert vitamin D to alpha hydroxycholesterol, which is the active component, and many of us are lacking or low in vitamin D, suggesting that there might be nature's ways of basically maintaining health. And while methods to predict, protect, precision, all of them are super important, maybe we should go back to what nature has told us, comparative biology, what human physiology tells us, to try to identify intervention points. So I'm going to stop there. So what I've tried to tell you is give you a few examples of how we can sort of build on mechanism. I've touched upon very different topics, and at the end I've basically used cholesterol as an example of going from an observation based on a lab bench to a very curious graduate student to finding that the Framingham Heart Study confirmed our findings. I'll stop there and I'm happy to take questions.
Thank you, Ramnik. That was fantastic. Now, one of the immediate questions is can you manipulate the microbiome in a sort of intentional way rather than it just happening in order to achieve benefits like this?
Yeah, so the cholesterol story is turning out to be super interesting. So cholesterol in the body is absorbed by these NPC receptors, transporters, that are found in the small intestine. So we asked the question, where do these bugs live? They live in the same region. So they compete for the same substrate. And basically, it's a battle between, does the bacteria get hold of the molecule or the transporter get hold of the molecule? And if bacteria can convert it, then you excrete. So the hope is that we could either deliver the enzymes in a pill that gets released in the mid-jejunum to distal jejunum, or we could find ways of enriching for a location-specific microbiome. So it's turned out to be, we have a paper that's just recently been accepted in Nature, where we, in another study, one of the students mapped every single transporter in the human body, all the way from the duodenum to the colon, diet, GPCRs, how circadian rhythm alters expression. So I think these types of maps combined with physiological measurements, I think could play an important role.
And one of the lessons from that is that the microbiome you're talking about is not just in the large intestine.
Correct.
Yeah, and the small intestinal ones are important,
probably
especially if you're going to generate vitamin D metabolites.
Yeah, I had a story on vitamin B that I took out because I was worried about time. There's another fascinating story where the vitamin B is made by this choroid biosynthesis machinery, and choroid biosynthesis, so the bacteria that Mishitaga at Berkeley studied for many years, couldn't really do it. And so we now found another bacteria that can hand over an enzyme to the bacteria that Mishitaga was working on by horizontal gene transfer. Mm-hmm. And now that bug can make all the vitamin Bs.
Questions from the audience? One at the back and then at the front here.
A quick question, Ramnik, on intervention potentials. And my question is more related to the whole complexity of the ecosystem of the bacteria, and how do you see the science progressing in manipulating one bacteria and not disturbing the totality of the ecosystem? Yeah, so
potentially good part of the cholesterol story is we know where the bacteria are. The second thing is that it's its natural habitat, and so if we've done these experiments where we've fed it on its own, depleted it, and it hasn't changed, at least in mice, the small intestinal microbiome. So you can basically add, but I think the clever strategy is probably going to be, now that we've isolated the enzymes, to give the enzymes in a capsule form.
Okay.
Why the enzymes and not the bacterium directly?
Yeah, so you could do both.
Or maybe transferring the enzyme into a probiotic type of system.
Yeah, so so there are lots of ways of doing it. We could give a probiotic. We could find probiotics that are currently used and engineer the enzyme into it. But I think one of the things that we've been broadly thinking about is how to design a clinical trial that's not going to run up against EU restrictions and the FDA. And so what we want to try to do is the simplest entry point. And what's striking is now with the Framingham Heart Study, we've now got enzyme work, the 4th cohort that's now being studied. And we can now get a second measurement on the first patients, and it's 100%. If you had it, you have it now.
Okay. Ryan and then Kartik.
So, Ramnik, thanks. That was an excellent talk. I'm going to move away from Oscillobacter to something you shared earlier in your presentation on the microbiome molecule mechanism. So, clear on the piperacolic acid. Have you done any work to understand the downstream mechanism of piperacolic acid's mechanism on length?
Yeah, so there's an elegant study that in Drosophila, if you basically give piperacolic acid, the Drosophila lengthens. And they've been able to identify the enzymes and the signaling pathway that's behind that lengthening process. A student in my lab now hasn't nailed it yet, but is very close to figuring out. So we know what the enzyme is. We want to knock it out in the bifidobacter that I showed you, and showed that this bacteria without the enzyme cannot make piperacolic acid. But the genetic data based on the Drosophila literature is pretty rock solid.
Kartik?
So, that was a great talk. Do dietary interventions like high fiber diets change Oscillobacter levels?
So we haven't looked at that. There could be ways of enriching for these bacteria in the small intestine. Most of the microbiome metagenome studies to date are done on stool microbiome that largely captures the colonic microbiome. You can computationally figure out ways of identifying based on abundance measurements, which bacteria come from this place or the other. So I think these are studies that could very easily be done going forward.
Stefan?
Coming back to the cholesterol topic. Now, I was a bit surprised when you said, well, the effect size is equal to stanols, which you get on the market, because, my understanding is that cholesterol levels are not largely impacted by dietary cholesterol. Of course, you have some change. So if you cut totally the cholesterol intake, well, you get maybe a 10-20% reduction. But what about the rest?
So there is this enterohepatic circulation. So bile salts get recirculated back, and bile salts are built of cholesterol. So when the bile salts come and get dumped in the terminal ileum, if this bacteria and enzyme is there, then you can now convert that into a non-absorbable form. So you now target a lot more than the 30%, because you also can convert.
So in fact, you're not targeting only the dietary cholesterol. Yeah. You're targeting then the total cholesterol.
The enterohepatic circulation.
Thank you.
And that's essentially the way the statins work as well.
Correct.
To break the enterohepatic recirculation. The aging and super aging was, what is triggering that change that you observed, especially in the very old? Is it part of the aging process? Is the gut function changing and influencing the microbiome?
So there are lots of unanswered questions there, but as Andrea was presenting, so I have a little bit of an advisory role in Singapore right now, and to the largest sort of research program in Singapore. And one of the fashionable things right now in Singapore is to basically have these luxury homes for retirement in Malaysia. So people basically go there, they have a healthy living standard, and hopefully the clocks are going to expand. And I'm sure this is happening in many parts of the world. But what's striking in Okinawa is that the 112-year-old, the 15-year-old, they go out every day to their garden. You know, they garden their small plot, they collect their flowers, they collect whatever, so that physical activity is an important component. So I think it's, and that's why I put that earlier slide about circadian rhythm, exercise, physiology, diet, microbiome, all of them have to be integrated in one collective unit. But there is a lot more that could be done.
Any final questions? Brian? Okay, last question.
So I just have a question. In your Okinawa population where you showed the lithocholic acid profile, do you have the primary bile acid profile there of
Yeah.
those?
Yeah.
And any interesting findings there?
So
that
the primary
drive
bile acids don't change.
They don't change.
Yeah.
Okay, thank you.
Yeah.
Okay.
Okay, thank you. Yeah.
Okay, and the second and final presentation of this section is from Alexander Mathys from Zürich. Although if you look at his CV, you'll see that he was an employee here at one stage in the development of his career, and he's going to change topics slightly and talk more on the engineering, food engineering side of things, intersection of food systems and nutritious diets with focus on affordability, which is one of the critical things I think going forward. So Alex.
Yeah, thank you so much, Ian. Hello, everybody. Thank you so much for the opportunity to present our work from Zurich and Singapore here at the Nestlé Nutrition Symposium. Very glad to be here. And as Ian already introduced, I want to discuss with you a broad field: food system sustainability, nutritional quality, and affordability. So how that is possible in 25 minutes, I think this is not really possible in a systematic way, but I want to focus on certain aspects which can be of relevance. And first of all, I want to clearly make clear that if we want to work on food system sustainability, we need to have a holistic view. We need to team up. Collaboration is key. And I was happy to be part of the Nature Sustainability Expert Panel where we could collaborate with social scientists, economists, agricultural scientists, food engineers on food system transformation, agri-food system transformation, where we need to have a bundle of social, technical innovations. So we need to combine our technical and biotech innovations with social innovations. How can we maybe give an example? Food waste, for example, the major challenge in food waste is not how we use it the best way. The major objective would be to avoid it, of course. It would be a behavioral aspect, or to reduce it would be also a behavioral aspect as first two priorities before we come with biotech and technological solutions. However, I'm a food engineer, so I want to give a bit the perspective of an engineering side. And if we see the challenges in these reports, 200 pages on the right, we can say, okay, 14 million square kilometers of arable land we have available on our planet, which is not infinite. We can't grow forever. Adam, you said animal-based food products will be more consumed. I agree. In the past with GDP, clear correlation, but there is somehow a supply limitation. So we can't produce more food on arable land because we need to increase it. It didn't increase in the last decades. We can, of course, use more plant-based food instead of giving it to our animals. Currently, we farm 19 billion chicken, more than humans on the planet. We slaughter 69 billion chicken annually. According to the World Economic Forum, we farm 1.5 billion cattle, 1 billion pigs. I think this is too much. But of course, there is a demand side. With increasing GDP, there is a demand for more animal-based products. I'm curious to see how demand and supply will develop. So what can we do? We can cut the rainforest. I strongly recommend not to do it, to increase the arable land square meters. We can produce more per square meter, and we've done that in an impressive way in the last decades, mainly driven by massive use of fertilizers. Haber-Bosch process is here one of the key innovations from the last 100 years, let's say. However, we use already 50% of our primary energy, that is 10 exajoule of the primary energy alone for fertilizer production. So I think there is a kind of high productivity already given. So we focus now on systems on non-arable land. which can be produced on non-arable land, animal-based products, but also microbial-based products. And I want to focus a bit in this talk on these aspects. So if you work on such a complexity, I strongly recommend to collaborate. I'm glad to be in the Institute of Food, Nutrition and Health at the ETH Zürich with a lot of great colleagues, which are partially also here. Patrick from the engineering side, Ferdinand from the nutrition side, where we have joint positions. We have the chemistry on board, the microbiology, the biotechnology, and also the consumer behavior. And I will come to certain aspects of these collaborations, by the way, which is not only internal, we collaborate also with a lot of external partners on these aspects. But coming now maybe a bit more specifically to food system sustainability. Sustainability has 3 dimensions. Very often we discuss only the environmental sustainability, which is, for example, even having ISO norm methodologies like life cycle assessment of a product or service, which gives us the total environmental load, for example, of a kilogram of food. So you already hear a kilogram of food, so what? Is there a nutritional quality considered? No, it's not, because nutrition and health is part of social sustainability. It's not quantified very often in environmental sustainability assessments. That's why we suggested this study here in 2018, combining 25 sustainability indicators in 7 domains. We analyzed the food consumption in 156 countries and suggested these 7 domains, such as food nutrient adequacy, including different nutritional quality indicators, ecosystem stability, so the classical environmental sustainability portfolio, but then we move already away to social and economic sides, affordability, which I want to discuss a bit more in detail today as well. Availability, is it available? When it's not available, it doesn't matter whether it's affordable or whether it's nutritious. Social culture well-being, resilience, food safety, and waste and loss reduction. You can expand this system, but it gives you a little flavor what it means when we focus on a holistic sustainability view, and here you can identify certain opportunities and trade-offs. Today in this talk, I want to focus a bit on the high environmental impacts linked to animal-based products production, specifically the animal-based protein-rich foods, which give a lot of important nutrition if we consume them in a reasonable way. Many of the substitutes which are currently on the market and discussed maybe have a similar structure, maybe a similar taste based on food engineering innovations, but I argue is it the same nutritional quality, especially when it comes to bioavailability, and that was a strong focus of yesterday's discussion as well. I think we can improve a lot. And whether they are really affordable, very often affordability, let's say the lab meat, we are far away from affordability for a world population or for an impactful integration of such a solution yet. Here I want to suggest a biotech innovation focusing on polyextremophilic organisms which can reduce production cost, can suggest protein-rich, nutritional, high-quality, nutritional, protein-rich food alternatives, and hopefully can be integrated in our food system also on the global perspective based on different regional needs. And I want to go on a bit on this journey with you. So I mentioned regional needs. When we quantify nutritional quality and environmental impacts, there are data available for country-specific environmental impacts, for example, published by Dr. Poor and Dr. Nemecek, our partners here in this study, where we could then really quantify more than 180 countries, the environmental impacts of different food groups and food products. We combined that with Adams' nutrient-rich food index, NRF, in this case 21 beneficial and 2 disqualifying nutrients of the different foods classified in groups, and could then see really in different regions how the nutritional quality and the environmental impacts are changing. We call that nutritionally invested environmental impacts. So sometimes it's worth maybe to invest a bit more environmental impacts to have a higher nutritional quality. Vice versa, we can also identify micronutrient needs in different regions and then try to compensate these micronutrient needs, for example, with foods which have a lower environmental impact in this specific region. For example, where we don't have too much water stress or where we have maybe more land use available. Here we used country-specific databases like the Global Expanded Nutrient Supply Database, the GENUS database, and as I mentioned, the data set of Poor and Nemecek from the Science paper 2018. Affordability, very often mentioned in the last 1.5 days, how we can maybe improve affordability. That was often said. It has to be affordable. I won't focus now on the social aspects of affordability. I want to focus on technical aspects regarding affordability, specifically focusing on capital expenditures, operational expenditures, and also innovation readiness level, because we talk also very often about new solutions. I don't talk now about the indirect cost of a product, which we can quantify by life cycle costing, showing, for example, hidden cost by environmental damages of the production. This is excluded in my focus because I want to first focus on the direct costs, which we have better under control. Capital expenditures, that's the factory, for example, you have to build up. It has to be a scalable production very often, otherwise you can't reach economy of scale. Operational expenditures, that's, for example, the substrate you need to have for your production, the fertilizer, for example. And the innovation readiness level is relevant for solutions which are not highly optimized yet and scaled very often. And here we talk about customer readiness level, team readiness level, business readiness level. IPR stands for IPR stands for patents, for example, for intellectual property rights readiness level, funding readiness level, and last but not least, the most known readiness level, the technology readiness level, which is shown here. That is a level which was introduced by NASA for rocket development. We use it a lot in the European funding scheme, Horizon 2020, also for our project status reports. You can see here from a TRL1 on the x-axis, from the discovery in the laboratory by your students, or hopefully also you joined that discovery as well, to a TRL9 where we demonstrate industrial feasible production in an factory environment. That is a long pathway. It can take quite a long time and quite significant resources which are represented here by the red line. And you can see in the beginning we are wealth. We get a lot of support from pre-competitive funding, from the taxpayer, for example. Then there is a drop because in food processing and engineering you develop something then to a pilot scale, 200 kilograms per hour. You demonstrate feasibility on the TRL 5 and 6, and then everybody's saying, okay, job is done in academia. Please, industry, take it over. When I then come to Stefan, who is asking me, Alex, we need to have tons per hour, otherwise we can't integrate that. So please continue the development. And this is really often a challenge, which we, for example, need to do then with small and medium-sized enterprises, spin-offs from the ETH, for example, and groups which are supporting the next generation then from pilot to industrial scale. And then we have the economy of scale to really cut down the cost to a level which is delivering affordable products. So now I want to present to you some two case studies from biotech innovation focusing on polyextremophilic organisms, which are part of this value chain approach in our laboratory, in strong collaboration with our partners, which deliver alternative feed, because I believe we need to have less animals on our planet, but we still need animals in our agricultural food system to not only run the agricultural production, but also to deliver bioavailable micronutrients, for example. And I want to focus on some extremophilic microorganisms, specifically microalgae, to deliver interesting new protein-rich food products. But let's first maybe start with the feed. Here we focus on residue utilization, which can't be avoided. That is, again, the first objective. Can't be reduced. And then on the third priority, we can give it to our extreme black sugarfly larvae system. You can see a production facility here on the right in Surabaya in Indonesia, which was run by our partner, the EAWAC. We also collaborate here with colleagues in Kenya, Nairobi, Sanergy, and in Rwanda, where we can run such a low-cost, let's say, a relatively low-cost facility. You can see here's a plastic crates with a species which is very tolerant to very heterogeneous substrate intake, which we can collect from markets, for example. Here in Surabaya, we take the fruit and vegetable residues. We can take food waste, which we do in Singapore, for example, in our colony. And we take also other side streams here in Zürich with the EAWAC Aquatic Research Center. You pre-process it, then you add the larvae to the waste. The larvae reduce the waste, 50% or even more. So you have also a waste reduction performance. And then you separate the larvae from the residue. The residue can be used as fertilizer, which has an antimicrobial activity. The larvae contains protein and fat, which can be used for aquaculture feed. 5 million tons globally are produced by, fish meal is produced globally for the international feed industry, and very often I observed in developing areas there is a lack of supply of feed very often, and of course also then prices went up. So here maybe also for decentralized productions, farmers can run a little colony with the biotechnological innovation presented here. The products look like this. You have the protein meal, up to 40% protein concentrate. The insect lipids are separated before the protein is concentrated, because otherwise it would be clumpy a bit, and the fertilizer is a side product. And this can be an alternative feed, supporting a lower number of an animal-based production chain, maybe also in developing areas, suggesting alternative feed to fish meal. We investigated in terms of sustainability performance this production in 2018 of the most advanced production facility in the Netherlands. And here you can see a comprehensive life cycle assessment. So we quantified the environmental sustainability only of 1 kilogram of puree on the left, or 1 kilogram of protein concentrate on the right. You can see here the bars are going down. The y-axis is demonstrating the total environmental load. You can see all the environmental indicators on the in the legend, which I won't go in detail now. It was an isonormed investigation. And we could demonstrate in the scenario development that when we can improve feed efficiency by new breeds without GMO, when we can use non-utilized side streams as a midterm scenario, and finally as a long-term scenario, change completely to renewable energy, we could significantly reduce the environmental load of the production of 1 kilogram of insect-based feed. That is demonstrating the potential here of environmental sustainability performance, because this is, in the end, the main driver why we want to go for insect-based feed. But now switching a bit to the food side, here we focus on an extreme microalgae, also other microalgae. We mainly focus a bit on fermentation, not on the classical microalgae autotrophic cultivation in the outside environments with photosynthesis. We believe in mixotrophic and heterotrophic cultivation. And I want to show you some interesting aspects of a new species, which is called Galdiera suferaria. It's short before novel food approval, for our point of view, was submitted by a partner of Givaudan, partnering company. It can have up to 64% protein with very diverse micronutrients portfolio. Also, depending on the cultivation, you can change the nutritional composition of the biomass. It grows at pH 1. Which is for me an absolutely outstanding element of this production, because with that I can save decontamination costs. I don't need to work in sterile conditions, a big problem in fermentation in general, because then I need stainless steel and then I have too high capex cost. It can run up to 56 degrees C. Another strong cost driver of outside cultivation, especially in subtropical or tropical areas, is the cooling. In subtropical areas, maybe sometimes also heating. We can run it in continuous mode. This is a strong benefit as well. We run it in Zürich over 3 months, so we can have a continuous processing and production. And in general, the food industry is looking for continuous processing to reduce cost with high productivity. We can have circular residue stream utilization. We now investigated more than 50 residue streams, and the species seems to be somehow accepting it. It's not the best performance always, but we can run heterogeneous substrate input. Substrates are a strong cost driver of many fermentation processes. We can run it in mixotrophic oxygen CO2 balanced cultivation. I will explain that maybe afterwards a bit more. It's a very specific aspect of this species where we don't need to inject gas. Gas is also a cost driver. We need to have a pressurized tank next to the cultivation very often. And of course, the gas needs to be also paid. So we can avoid that. And that is leading to a simplification of production of a protein-rich food-grade biomass, which is now self-affirmed grass by the company, which is using it, but we also use different strains, and we can produce bright colors, which are also a strong importance when you want to produce food that you don't have only dark biomass. You can see here our production asset, which we investigated in Zürich with a 30-liter reactor, so we could produce enough biomass. And then the question was a bit, and now I want to connect also with the discussion yesterday, where I also asked a question. What is now the key performance indicator when we produce biomass? Is it the protein content, if we want to produce a protein-rich food category, finally? Or is it the bioaccessibility? Now in fermentation, we work with microbes. This microbe has a protein-based cell wall. The bioaccessibility of the highest protein content biomass was only 14%. The other species, the other strain, with 51% protein, had a much higher bioaccessibility, 62%. Currently, we investigate bioavailability, also in strong collaboration with Professor Ferdinand von Mayen. So we need to relate a bit. What is the nutritional target here? On productivity, we could have a 2.3 times higher productivity than the classical autotrophic cultivation, which you know when you see the tubes outside the buildings when you produce with photosynthesis only, with mixotrophic cultivation, where you can increase significantly the concentration, you can have a much higher productivity, again, reducing cost. On top, we now found out that there is a very high amount of micronutrients of interest in the protein-rich biomass. So we don't deliver only proteins, we can also deliver iron, zinc, with huge amounts like 1,479 milligram per kilogram. I hope they are not all bioavailable or bioaccessible. We need to investigate that. That is running R&D, and we hope we can publish that soon. Also, zinc was available, and we also now investigate other micronutrients of interest together with our colleagues in ETH. Yeah, so what can we now do or summarize when we see this quite extreme biotech aspects, and we talk here about non-GMO? GMO gives another dimension, and we are open for that in general, but it has also a lot of limitations. We talk here about regulatory and safety hurdles on top. This is all so far non-GMO. We can reduce CapEx when we produce a higher nutritious biomass because it has certain functionality. Functionality gives value. Techno-functionality gives value in terms of structural benefits. We can structurize multi-phase systems. We need to structure food to have consumer acceptance. Biofunctionality is, for example, bioaccessibility, bioavailability, and bioactivity. And if that is given and proven with evidence, then we can justify investments and operational expenditures. Flexible cultivation can be adapted to regional needs. Think about the regional study I've shown to you. There are food compounds with different nutrient contents in different regions. There are different nutrient needs in different regions. There are different availabilities of certain residues, and then we can adapt our cultivation if it's flexible enough, if it's robust and resilient enough, with the different substrates which we can use. We can homogenize them. However, and this is a recent publication with the National University of Singapore in our laboratory there, if you use residues, you can't just cut the cost for the substrate, which you normally would buy. You have to also add CapEx for the hydrolysis line. So that is an engineering aspect which shouldn't be forgotten. So that is investment and also energy which goes into that. However, from my point of view, it's always better to avoid material cost when we have a continuous processing line which runs hopefully then for 5 till 20 years. It can grow at pH 1 or 2. With that, we don't need sterile conditions. We didn't observe any contamination with our continuous cultivation over 3 months yet. That gives a lot of opportunities, reducing cost, and that also specifically runs simplified reactors. We now built up the first simplified reactor for Galdiera worldwide in the ETH Zürich, which costs less than 500 francs for a 50-liter reactor. We hope we can push that further and also introduce it to developing areas. We can run it without cooling, a big benefit in subtropical and tropical areas. Cooling is a big problem. When it goes too high with the temperature, quite often microbes have an issue or even die off. And we can run it in oxygen CO2 balanced cultivation. This is a very new innovation from our biotechnological team lead, Dr. Fabian Abusi. What else we can do? We can also add pure technology, and of course I need to talk about pulsed electric field. It was mentioned yesterday and Kieran is laughing. What I want to show you here, technology itself, normally when it's a lot of stainless steel, it has its limitation when we want to introduce it in developing areas. However, here we have a technology on the left which can stimulate microbes. It's a stress inductor. So we use only 500 joule per kilogram, less than 500 joule per kilogram, and we could observe up to now 40% more biomass after 5 days of cultivation. This can be a game changer for several fermentation. However, we don't reach more biomass than the benchmark, we reach it faster. So we reach the ultimate concentration maximum faster. We can also milk cells with this technology. You can open cells with this technology and close it again. They stay alive, extract nutrients. That is interesting in the continuous cultivation. Maybe not at pH 1. We have other strains which operate at pH 7, but this is currently under investigation as well. And we can disintegrate cells with much less energy. You can see a little picture with my first doctoral researcher from ETH Zürich. It took us 5.5 years to commercialize this new nanosecond pulsed electric field cell stimulation system, and it runs now with Bühler with 20 tons per hour for plant cells. So this is really industrial feasibility and scalability. What else we can do? What do we do with the final biomass? Yeah? So I talked now a lot about primary production. Can we translate that into food products? We can translate that into protein-rich alternatives, maybe to animal-based meat. And we combine it with plant protein concentrate, because otherwise it wouldn't be affordable from my point of view. Let's combine the good things from plants and microbes, and then we can generate structures. And this wouldn't be a green meat substitute, it would be really a bright one, which can be then modulated in terms of taste when we add some flavors from our colleagues. This has a neutral taste. And on top, it has a higher nutritional value for our point of view. We could, just by adding microalgae, fermented microalgae, add increased vitamin E, B1, B2, B3, and B6. Even after extrusion, it was measured. Is it bioaccessible and bioavailable? I need to ask you to be a bit patient because this is currently under investigation. With that collaboration with Ferdinand von Meyer's laboratory, which I'm very glad that Ferdinand and our laboratory have joint positions now. So the food engineers have a joint postdoc in the nutrition lab and vice versa. And I like that very much. We can work on bioaccessibility with its limitation in vitro, on bioprocess optimization, and then go to bioavailability ex vivo with KQ2 cells and also in vivo to mice and also human intervention studies where we focus currently on iron and zinc from microalgae, which looks very promising. Yeah, however, and this we heard very often, and Kieran always brings us up, also of course Stefan in discussions, if nobody wants to have it, so what? Insect-based food was a huge initiative and hype a bit maybe also. I had a lot of hope, but there is still a huge resistance. For microalgae, we have a better opportunity. Here you can see a consumer study of 578 Singaporeans, which link microalgae-based meat substitute to innovative elements, environmental friendly and healthy. It was a bit neutral regarding nutrient-rich and high in protein. I expected a bit these benefits more pronounced. However, we have a relatively positive view, which is very the opposite to, for example, insect-based food, which I didn't present today. On top, regulatory is now opening up more and more strains, and as I mentioned, Galliera zoforaria, as an extreme case maybe, will be approved hopefully end of this year. At least that is our strong expectation. With that, I hope I could summarize a bit what is our approach in terms of multi-indicator sustainability assessment, adding nutritional quality, affordability into the equation, having case studies where we try to suggest feed and food innovations based on non-arable land production systems, because we need to produce 60 till 70% more food in the future, according to FAO. All our arable land is used. And with that, I would like to summarize the take-home message. So let's do holistic sustainability assessment. We have methodologies available of all three dimensions. And we won't be best in class in all. Let's identify opportunities versus trade-offs in different regions. And regions have different needs. You can't generalize that. We can produce alternatives to nutritious and affordable, or we can produce nutritious, affordable, and more sustainable protein-rich foods, from my point of view, based on the suggested solutions. Affordability of emerging foods is often not given. It's discussed. Maybe the nutritional elements are given, but then it's by far not affordable. CapEx and OpEx reduction are absolutely needed. And we have to focus a bit here less on stainless steel, but because the CapEx are significant over the depreciation charge of such a solution. Functionality gives value and justifies investment. And I hope I could explain you why I'm so excited about polyextremophilic microalgae and polyextremophilic insects, because you can reduce these costs. And I suggest a fair comparison. That's still new solutions. If you compare it with soy production that was optimized over the last 60 years, we have a very high technology readiness level, innovation readiness level, and the economy of scale. We still need to do that. But if we recognize the readiness level with the existing state of the new solutions, I think then there is a fair comparison possible. And last but not least, let's iterate with holistic sustainability assessment. I would like to thank our team in Zürich and in the Singapore ETH Center, where we run a food translation laboratory, all our partners from academia, R&D centers, spin-offs, startups, and multinationals. And last but not least, I thank you for your attention.